Thomas Turbyville, investigador principal del Frederick National Laboratory for Cancer Research. Cuenta con una carrera de más de 12 años trabajando como investigador de cáncer con amplia experiencia en microscopía. Su investigación se enfoca en el uso de microscopía avanzada para visualizar los procesos complejos de transformación celular. Participa activamente en la identificación y caracterización de moléculas con potencial terapéutico y la definición de sus mecanismos de acción. Con más de 20 artículos en diversas áreas de investigación del cáncer, el Dr. Turbyville destaca como un investigador clave para el avance básico y tecnológico del Laboratorio Nacional Frederick para la investigación del cáncer. Recibamos con un caluroso aplauso a este gran investigador. Él es Thomas Turbyville.
Muchas gracias. Me encanta estar aquí en Puebla con ustedes. Gracias muchísimo por la invitación para atender esta reunión. No voy a hacer mi presentación en español, pero nomás quería darles un gracias por invitarme aquí. Bueno, in English now. At the Frederick National Lab, we do team science, and our focus is on RAS. RAS is a major oncogene in cancer. It affects about 30% of cancers. And really to solve the problem of RAS, which has been studied for 30 years at least, you need a lot of skills, a lot of technologies, a whole variety of approaches in order to be able to understand it, and then also to develop therapeutic approaches to targeting it. So I'm going to talk about one piece of that effort, but I just want you to know that there's at least 60 or 70 scientists who are all working together as part of a big team. And my mentor and boss, Dwight Nisley, is going to talk a little bit more about that later on. You'll hear more about what we do. So as I mentioned, we work on RAS. KRAS is the particular isoform that is most commonly mutated in cancer. And you can see this is just in the US population. For 3 mutational types, or 4 rather, there are about 100,000 patients per year that are impacted by this mutation. And there are no therapies for patients who have mutations in KRAS. So most of these patients die. So we're talking about 100,000 people a year in the US alone, probably about 1 million people worldwide who are suffering from cancers of the lung, colorectal, and pancreas from mutations that are driven by KRAS. KRAS is a signaling molecule. It functions like a light switch, essentially. It goes to the plasma membrane, and it is switched from on to off by loading with GTP, and then hydrolyzing GTP to GDP, it's switched to off. And in cancer, it gets stuck in the on position, essentially. So the mutations cause it to no longer be able to hydrolyze GTP through the effector interactions with the gap proteins. So you can see here that there are a lot of pathways that are impacted by RAS downstream. And many of those pathways are implicated in cancer. Just proliferation, for instance, is driven by RAS. So when RAS is mutated in the on position, you are constantly getting proliferation signals in the cell. Pancreatic cancer, many of you I'm sure are familiar with pancreatic cancer. It is an especially deadly form of cancer. It's relatively rare compared to lung cancer and colorectal cancer. But the patients who have this disease, almost all of them have KRAS mutations. And in fact, it's probably one of the first things that happens in the natural history of the progression of cancer in the pancreas. So KRAS mutations are probably the driving and cause of pancreatic cancer. And unfortunately, patients who have pancreatic cancer do not do well. The 5-year survival rate is only about 5%. So out of 100 patients who have pancreatic cancer, only 5 are still alive at 5 years. So this cancer is a very important target, or this mutation in KRAS is a very important target for understanding. And so our efforts at the Frederick National Lab are focused on really understanding RAS better, using a variety of approaches, and then also developing therapeutic targets or ways of targeting RAS that are new and relatively unexplored. So, as I mentioned, RAS is a membrane-localized protein. On the right there, you can see confocal images of two different isoforms of RAS. So the RAS are tagged with green fluorescent protein, and you can see that RAS goes to the plasma membrane. And if you look at these two micrographs closely, you can see that there are subtle differences between KRAS and HRAS, which is another major isoform of RAS. So the localization of these molecules in the cell is slightly different. Over here on the left, you can see why they're different. So the hypervariable region, the C-terminal end of RAS, all RAS isoforms, is very distinct for each isoform of RAS. And there are subtle modifications that happen on the hypervariable region that act kind of like a zip code for directing where RAS goes in the cell, in the plasma membrane. KRAS in particular, in its hypervariable region, has a bunch of positively charged lysine amino acids that then form interactions, stable interactions with negatively charged lipids in the plasma membrane, like phosphatidylserines. So those interactions are slightly different than HRAS and the other isoforms. And that actually represents an opportunity. The way that KRAS goes to the plasma membrane is different than the other isoforms of RAS. And so you can imagine that a small molecule, a drug-like compound, might be able to inhibit that transition from where it's folded on the ribosome in the ER to the plasma membrane. So those pathways that you're seeing over there on the left of where RAS goes in the cell all represent potentially different targets for RAS drug intervention. And in fact, people have used this approach more or less to try to target RAS. So farnesyltransferase inhibitors were a series of drugs that were developed 15, 20 years ago that showed a lot of promise because they inhibited the lipidation of that hypervariable region. So there's a farnesyl group that's attached to the C-terminus of all RAS molecules. And that lipid tail is how it inserts into the plasma membrane. And so they found drugs that inhibited the enzyme that puts that lipid tail onto RAS. And they thought, oh my gosh, we have the cure, because now we can prevent RAS from getting to the plasma membrane. Unfortunately, KRAS4B, which is the isoform that causes most of the cancer, can also be geranylated. So it had an alternative path, which it exploited immediately in patients. So patients who were given these therapeutics did not receive any benefit from those drugs. And so unfortunately, there was a lot of disappointment about being able to target RAS this way. But it gives us a tool in order to develop our own assays for looking for other drugs. So right here, you can see confocal micrographs again. So these are untreated cells on the left. And then you can see with increasing concentrations of the farnesyltransferase inhibitors, there's less and less signal at the plasma membrane. There's less and less KRAS at the plasma membrane, HRAS in this particular example. So we decided to develop a confocal-based imaging platform to screen for other compounds that inhibit the translocation of RAS from the cytoplasm to the plasma membrane. And then also maybe find inhibitors that would bring RAS off of the membrane. So how RAS gets on and off of the membrane is actually not fully understood. So there are actually different ways that you can imagine molecules disrupting this interaction. So the approach that we used was to develop cell lines, HeLa cell lines in this case, that express an inducible form of GFP-KRAS, an oncogenic version of it, KRAS G12D. And then we have a way of staining the membrane and also the nucleus. So we have 3 channels that we can measure quantitatively now. And you can see that the RAS molecules are all very nicely distributed at the plasma membrane, and that overlaps perfectly with the membrane signal. And so now we have a mask that we can create. Image analysis can then tell us how much RAS is in the plasma membrane. And so we miniaturized that, putting it in actually 384 well plates. And then we go using automatic microscopy to each well and take multiple images from each well. And we can put different compounds from libraries and screen for compounds that inhibit RAS membrane localization. And then we can quantitatively measure that membrane localization using an image analysis routine that we developed. So the image analysis locates each cell, it finds the membrane using the membrane channel, and then it quantitates the amount of GFP that is co-localized with that membrane mask, and then gives us an actual number about membrane quantity of RAS. And then if we have inhibition, we can actually measure that. So we developed a high-throughput assay that way, and then we collaborated with Eli Lilly, a pharmaceutical company I'm sure you've heard of it. It's a major pharmaceutical company, and we screened 20,000 compounds against this assay. We also developed other assays. So this is a protein-protein interaction assay. It's based on FRET, so fluorescence resonance energy transfer. This is a variant of FRET where you use bioluminescence. Instead of having a fluorophore as a donor, you have a luciferase as a donor. So you generate light energy, and that energy sometimes transfers if you have a molecule that's very close by that can absorb that energy. And so that energy exchange gives you a quantitative readout for protein-protein interaction. So you can see up there at the top, that's a cartoon. So you have RAS labeled with your luciferase, and then you have the effector that interacts with RAS that generates the downstream signal, RAF. A little confusing because they're very close in RAS and RAF. I often get those switched around. But anyway, the RAF molecule comes from the cytoplasm and binds to RAS. And when it gets that close, there's an energy exchange. And we actually know very precisely the molecular details of those interactions because we have crystal structures of the effector binding domain of RAF along with KRAS, and we know which residues are responsible for those interactions. So we can mutate those specific residues in our probes, and we can develop control. So you can see down here in this graph at the bottom, this line graph, there's a saturation curve here. The line in green is KRAS G12V, so oncogenic variant, along with a full-length RAF that has a mutation in the RAS binding domain of RAF that prevents that interaction. So that's a control that tells us, yep, we can actually see no interaction when we express these probes, but then when we combine the oncogenic variant KRAS Q61R, which is always on, we see a lot of interaction with RAF. So that gives us the basis of an assay. This is our signal window, which is actually about 15-fold. So using that assay, we screened the same 20,000 compounds that we screened with Lilly on the localization assay. And this is just an example of the sort of data that we get out. So you can see that most of the compounds that we tested are in the middle, those colors in the middle, those are inactive compounds. They're not affecting the interaction at all. But you see some dots that are inhibited, that are low. Those are molecules that actually disrupted the interaction of RAS and RAF. And then once we did that single point assay of 20,000 compounds, we then went back to the actives and we did dose response assays for all of the actives, and we further validated the ones that were active from the original 20,000 compounds. So we started with 20,000. We found 178 inhibitors in the primary screen. We also found enhancers, so molecules that cause more interaction. And then we did dose response assays, and that eliminated a lot of false positives. And then we did a counter screen with a FRET control. So that's just looking for molecules that are actually just inhibiting FRET that has nothing to do with RAS. And so then we got down to about 35 compounds that were actually positives. And those became sort of our lead compounds. And then Lilly went back into their library. So they have huge libraries of compounds, millions of compounds. And they looked for molecules that were similar to these 35, and they sent those to us. And we did some further testing, and we now have found some low micromolar active compounds that inhibit the RAS-RAF interaction. We also have developed assays that are further downstream. So you can see here, RAS signals to RAF, RAF signals to MEK, and then MEK signals to ERK. That's a signaling cascade. It's a very simple cartoon version of actually what happens in cells. So we want to know what things are inhibiting RAS localization to the membrane, what things are inhibiting RAS's interaction with its effector RAF, and then also we want to be able to measure signaling downstream. So the movie went by really quick, but we have a biosensor in cells that we can express. So let me just go back through it. So in cells, you can see when we stimulate with EGF on the left that the biosensor leaves the nucleus. So that's a readout of activation of the pathway. And that gives us very interesting information because not every cell is going to respond in exactly the same way, especially when you apply very low concentrations of growth stimulus to cells. If you look on the right, trametinib is a MEK inhibitor. When we treat those cells with EGF, we don't see any activation of the biosensor, so the biosensor is stuck in the nucleus. So again, this gives us a readout of activation or inhibition of this pathway downstream. And the advantage of it is that it gives us single cell information. So we can quantitate how the biosensor is moving in and out of the nucleus at a single cell level, and that will give us a lot more information than just doing a Western blot, which is a sort of traditional way to look at ERK phosphorylation or MEK phosphorylation downstream of inhibition of RAS. So those are some of the tools that we're using for drug discovery. There are a lot more things that we do in terms of drug discovery, but these are more cell-based approaches. We also do biochemical and biophysical approaches for discovering inhibitors, and Dr. Nisley is going to talk about some of those things when he gives his talk. So we have these confocal images. They give us some resolution. I mean, we can tell about where the cell is locating the different molecules. You know, are they going to the membrane, or are they in the nucleus, or are they in mitochondria? But the resolution is not very high, even in confocal imaging. To really be able to study RAS at a single molecule level and understand what's happening with RAS at the plasma membrane, we need to be able to use techniques that allow us to see single molecules of RAS. Even super resolution, sub-diffraction levels of resolution. So the optics are inherently limited in how much resolution they can achieve. But in the last couple of decades, people have been able to develop techniques that allow you to localize a molecule to very high levels of precision. So sub-diffraction. So instead of having 250 nanometers of resolution, you might have even 20 nanometers of resolution. So that middle image there is a super resolution image of RAS localized at the plasma membrane. And you can see that there are lots of RAS molecules there, and there may be some organization to how RAS is sitting in the membrane. And people have been very interested in understanding whether RAS is forming dimers or higher order structures in the plasma membrane that are responsible for some of its biological activities. On the very far right there is a cartoon that was developed by a scientist from Japan named Kasumi, who's studied the biology of membranes extensively. And he has proposed a model for the plasma membrane that is very hierarchical. So you have, within the plasma membrane, lots of heterogeneity. You have protein interactions, you have protein-lipid interactions, you have lipid-lipid interactions, you have structure with the cytoskeleton forming a mesh holding up the plasma membrane. So there's a lot of complexity in there. And on top of that, the molecules in the plasma membrane are moving around. They're very dynamic. So the idea is that when a molecule comes to the membrane, it encounters open space at first, and then as it starts to interact with lipid and protein, it gets more and more constrained in terms of its mobility. And then it can start to form interactions with other molecules that then generate biology. So the membrane is not just a passive structure. It's very involved in the biology that's happening there. And so we have been using these single molecule techniques in microscopy and imaging to understand RAS molecules in living cells and then also in synthetic membranes that we build up in the laboratory. So we can make nanodiscs, we can make liposomes, and we can make supported lipid bilayers. And all of those afford us the ability to control the concentrations of the different lipids and protein. So that has some advantages. But we also really need to understand RAS behavior in the plasma membrane. So the setup that we use is a Nikon Palm system. It's called the N-Storm. So STORM is a variety of single molecule localization microscopy. And it's based on turf. So total internal reflection is just a physical property of light that when it encounters a surface that has a refractive index that's different than the media that's right above it, that light will completely reflect off of that surface. And that's indicated by that diagram there below. So when light comes in at a certain angle, that light won't go into the sample. It'll just reflect off of the glass cover slip. However, there is a little bit of an energy wavefront that goes into your sample, and it can excite the fluorescent molecules that are very close to the glass, to about 100 nanometers of depth. This allows us to restrict our view just to the plasma membrane, and we don't get a lot of background from the rest of the cell, which is above on the glass, 10 microns thick, which can create a lot of background. So we're able to get very low background images, and then we're able to use these very bright organic dyes that can attach to proteins. So they're permeable to the membrane, these organic fluorophores. They can go inside the cell and covalently attach to our protein tags that are on RAS. And because we have the combination of the microscopy and the organic dyes and then these constructs that we can express in cells, we can actually see single molecules of RAS in the plasma membrane. So you can see the overlay of the DIC image, the bright field image of the cell, with the single molecules of RAS moving around in it. And then we can then go into a very small area, about 10 microns on a side, and we can then observe single molecules of RAS moving around in the plasma membrane in a living cell. So using these single molecule approaches, and then also other techniques that we have in the lab, such as fluorescence correlation spectroscopy and other advanced imaging techniques, we can do biochemistry using purified RAS and lipids, and we can get a lot of information. We can extract quantitative parameters about the behavior of RAS, the oncogene and the wild type, the mutated version of RAS and the wild type version of RAS in living cells. And then we also work extensively with the Department of Energy. There are 3 national labs, Los Alamos, Livermore, and Oak Ridge, that have supercomputing capabilities. They actually have some of, if not the biggest computers in the world, and they run massive simulations of RAS and lipid, and we are able to then generate this very collaborative and mutually enhancing cycle of doing simulations, modeling, getting parameters out of the model that give us hypotheses that we can test in the lab, and then in the lab we can get further parameters that we can then fit into the models to improve the models. So as I said, we're using this total internal reflection microscopy combined with different versions of RAS that are halo tagged. As I mentioned, the hypervariable region of each of the isoforms is quite distinct. So as I mentioned, KRAS4B has all of those lysines in it, which carry positive charge. 4A, NRAS, and HRAS do not have as many lysines, and they don't carry those negative charges or positive charges on them. And they have other lipid groups that are attached to them. So each of those hypervariable regions is very distinct, and there's a lot of biology that we're trying to understand there, in particular with KRAS4B. So we can express these in cells, and then we can build up trajectories where we have precisely localized in each frame the location of the molecule to sub-diffraction levels of resolution, and then we can observe how that changes at a very high frame rate, 10 milliseconds per frame. So we can observe RAS diffusion in the plasma membrane and be able to extract parameters from it. And this is an example of some of the kinds of parameters that we extract. On the right there is a mean squared displacement plot comparing the different isoforms of RAS in a HeLa cell. The important feature of these graphs is the bend in the shape of the graph. If you look over here on the left, when you have a straight line, that is free diffusion. That's Brownian motion, so completely random motion. So if you see a molecule that's diffusing in the plasma membrane of the cell and it makes a straight line like that, you know that that molecule is freely diffusing. What's interesting is that KRAS4B, of all of the different isoforms, shows the highest bend in the shape of the graph. And that's an indication that those molecules are more confined. They're sampling a different environment than the other isoforms of RAS. And so we're extracting some quantitative information about the behavior of KRAS4B that is distinct from the other isoforms. And understanding what that means has been an effort that's been ongoing in the lab. So as I mentioned, these trajectories, if you look at them long enough, as I have and other people at the RAS Initiative have, you'll notice that there's a lot of interaction going on. And some of the molecules move very fast, and then sometimes you see molecules that are moving very slow. And sometimes you see molecules that are moving fast and then they slow down. So all of those things are happening, and it's impossible to see that by eye and quantitate that, but we have image analysis tools that we can use to quantify that. And one of those models is based on a hidden Markov model approach. So we can take the individual trajectories and analyze them and extract these models that show the different components or contributions of diffusion in the system. And what's interesting about this is that there are transitions. So the red circle are fast-moving molecules, and the blue are intermediate, they're sort of medium-fast molecules, and then the green are slow molecules. And in between each of those circles are arrows, and those arrows have different thicknesses. Those are the transition probabilities between each of those states. And so there's two things to notice. One is that the transition probabilities that go from red to blue to green are dark. And then if you look between the slow-moving and the fast-moving, they're sort of dotted, right? That's because between the green and the red, that transition happens only very rarely. It only happens infrequently, which suggests that there's a pathway that RAS molecules follow. They start as fast-moving molecules, then they go to an intermediate state, and then they slow down to more confined and immobile. And then they go back to intermediate and then to fast, not straight from slow to fast or vice versa. So to us, that suggested that there might be some kind of assembly process that we're capturing, or some kind of unique biology about RAS that we're capturing in our hidden Markov modeling. And when we compared the other isoforms of RAS, we didn't see the same level of confinement and the distinct mobility patterns. So if you look at HRAS over on the very far right, you can see that in the pie chart, it's only indicating two states. There's a sort of fast-moving and then a slower-moving state. It doesn't have the complexity or heterogeneity of mobility that KRAS4B has. This here is a map of the diffusion landscape of KRAS4B in the plasma membrane of a cell. So you can see on the left here, underneath the colored squares are trajectories. So those are the trajectories of RAS molecules. And then if you look very far on the right, you can see that those are color-coded. So when you see red or yellow, those are more fast-moving molecules. And when you see blue, those are slow-moving molecules. And when you make a 3D representation of that, it sort of forms like a landscape of mountains and valleys, right? So the RAS molecules slow down in certain regions of the membrane, and then in other areas, they speed up. And we suspect that there's a lot of biology happening in the slow-moving valleys of RAS molecules. This is just showing that when we put RAS in these mouse embryonic fibroblasts that only have one isoform of RAS per cell line, all the other isoforms of RAS have been knocked out, that we see the same kind of behavior. This was an important experiment for us to do because we know that the MEFs will not proliferate, these mouse embryonic fibroblasts will not proliferate unless they have an active RAS molecule in them. And this is telling us a number of things. One of them is that the tagged RAS molecules are physiologically active in the cell. They're generating a signal downstream that's driving the proliferation of these MEFs. And then we also see that the way that the RAS molecules are behaving in the mouse embryonic fibroblasts is the same as how we saw them behaving in the HeLa cells. Okay, important is the biology, right? I mean, if we just have mobility and we see these different kinds of mobility, that's kind of interesting and curious, but does it really have any biology underneath it? So these series of experiments are a little bit about the biology, exploring the biology. So on the very far right, we serum-starved those meths, and you can see that the serum-starved cells, those are very quiescent. There's no signaling going on in those cells. You can see that the line of the MSD plot, the mean square displacement plot, is more straight. So KRAS4B molecules are showing more random motion in the serum-starved cells. However, when you rescue those cells with serum, they start to bend. So when you activate signaling in those cells, those RAS molecules start to become more confined. That's, again, an indication that there's some sort of assembly process going on with these RAS molecules. In the middle there, those are mutants, okay? So we took the RAS molecules and mutated the switch-1 domain, which is the place where RAS interacts with its effectors, and we mutated it with a Y40C mutation, so that sort of weakens its association with RAF. And you can see in green that those molecules are more free diffusing. When you compare that to the oncogene Q61R, which is fully GTP loaded, always active, you can see that that molecule is very confined. It's showing more bend. So that again is suggesting to us that the biology of the diffusion, or the motion of those molecules, is related to the biology of the molecule. And then on the very far left, we took the G domain off. We took the G domain off of the protein entirely and just expressed the HVR. And you can see when you don't have the G domain, that KRAS4B in red is bent, as you would expect, but when you look at 4B HVR without the G domain, it becomes straight. So that again tells us that that globular domain that's responsible for the interactions with effector molecules is necessary for us to see this diffusion behavior. We explored the electrostatics. So we made mutations in the HVR. We mutated those lysines. We targeted them, converted them to alanine, or reversed the charge from a positive to a negative charge with the glutamic acids. And when we did that, we were able to see that not only did we change the diffusion behavior, so when you mutate those lysines to alanines or reverse the charge, the molecules start moving around faster, so the diffusion rate increases. But we also found that the residence time decreased with those mutations. So the amount of time that these molecules were spending, they're still going to the plasma membrane. There's enough of a signal for association with the membrane, but they're not spending as much time there. So the HVR is very important for these interactions. And as I mentioned, we have been working with our collaborators at the Department of Energy. They have done full atomistic simulations with using some of our parameters that we gave them to kind of model how RAS might be behaving in the plasma membrane. This is a very simple membrane system. We've just two lipid types, phosphatidylserine and phosphatidylcholine. And you can see when you run the movie that the RAS molecule, which is inserted into the membrane with its with the farnesyl group, you can see the little farnesyl group waving its tail inside the plasma membrane, that the G domain is actually interacting a little bit with the plasma membrane in this simulation. And so based on that simulation, we were able to make some predictions and understand some things about the behavior of molecules. So one thing is that we were able to see the clustering of negatively charged lipids around the hypervariable region. And when we changed the simulation, the hypervariable region changed the lysines to alanines, the distance in the simulation of the RAS molecule from the lipid bilayer increased, which is what's indicated in this graph over here. And then we did experiments where we depleted cells of phosphatidylserine using a genetic cell line that is a mutant that allows you to control how much PS is in the plasma membrane. We expressed the KRAS in those cells, and if we depleted the phosphatidylserine from those cells, we saw more free diffusion. So based on a lot of experiments that I'm not showing you, and a lot of further simulation and in-depth kind of biological experiments, we have a model that we've started to develop about what's happening. So we believe that what happens is that KRAS4B, when it comes to the membrane and inserts its farnesyl group into the lipid bilayer, that the positively charged lysines in the hypervariable region start to recruit negatively charged phosphatidylserine, the lipid, around the molecule, and that then creates an environment for the assembly of RAS into a molecular machine that can then signal downstream. But we're not there yet. We still have a lot to understand. And in order to understand really what's happening, we need to understand more about RAS's interaction with its effector molecules. And so we're using two-color techniques in order to see RAS and RAF at the same time in cells. So in green here, you have RAS molecules. You can see that they're pretty stable in the plasma membrane. And then in red, we have RAF, its major effector. And red is coming on and off of the membrane very quickly. And we're trying to understand in detail the quantitative features of those interactions of RAS and RAF in cells. So that's work that's ongoing to kind of further develop our observations that we learned using just one color, just looking at RAS. Now we're looking at RAF and RAS at the same time. So as I mentioned, we have this three-component system. So we have fast-moving molecules, medium slow molecules, and then slow molecules of KRAS in cells moving around. And it's a little bit of a mystery about what are the reasons for those three components. We don't fully understand it. And so to help us to understand that better, we're actually building up from the bottom. So instead of using cells, which are very complex and have a lot of other things going on in them, we're using simplified biochemical systems in order to try to see if we can recreate the conditions for seeing that three-component mobility. And so part of this collaboration with the Department of Energy has involved constructing a more relevant, more biologically relevant sort of lipid system. So instead of using just two, which is a lot of times what people do when they make supported lipid bilayers, they make these synthetic membranes, they use very simple components like maybe 1, 2, or 3 lipids. We're using 8 lipids. And each of these lipids is there for a very precise reason because it's representing some of the underlying biology that people have observed when they've done lipidomics of cells. So when they've extracted lipids from cells and done mass spec, they've looked at the relative amounts of lipid that are in the cell, both on the inner leaflet and on the outer leaflet of the plasma membrane. And so using some of those proportions and also paying attention to the charges on the head groups and also the saturation on the tails, and also including, of course, cholesterol, which is a major lipid in all of our plasma membranes, we have designed a system that's a little bit more relevant to actual biology. Keep in mind that in a human cell, there's often more than 200 lipids, 200 different kinds of lipids. So we're not getting anywhere close to that, but at least we're getting a little bit closer to the complexity. And so using that kind of approach, we then make supported lipid bilayers and put them on surfaces like mica in this case, and then use techniques like atomic force microscopy, which has a little cantilever probe that can, with atomic resolution, measure the surface of that supported lipid bilayer. And one of the things that we observe, you can see a reconstructed image. So the dark areas are deep areas of the place that was analyzed, and then the lighter areas are higher. You can do some profiling, so that line across, the blue line across the image is represented in a line graph, and you can see that you can measure with atomic resolution the depth of those different surface features. And that gives you some idea of like the lipid organization of our system. And so the important point to take home about this is that it shows that if you reconstitute a membrane using this complex 8-lipid system, you get transitions, you get phase transitions. And there are different domains, little small domains of lipid that organize. And that might be some of the contribution of the mobility of RAS. So when we look at the lipids, so these are fluorescently tagged lipids that are in our 8-lipid system. What you see is, based on the MSD plots, that most of those lipids, even though they're in that complex environment, are moving with free diffusion, so fast diffusion, random motion. This contrasts with when you put RAS in that system. When you put RAS in the system, 8-lipid system, you actually see more of a bend. So you're starting to see that confinement, the anomalous diffusion that I described in cells. So it looks like by using this more complex lipid system that we're starting to see some of the behavior that we saw in cells. So over here on the left, yes, you can see the same sort of graph that we made from hidden Markov modeling for cells, that when we do that for a simple lipid system, where we just have 2 lipids and we look at RAS diffusion, we really only see one component. We only see a fast-moving species. It's only when we have a more complex 8-lipid system that we start to see that three-component system. We start to see more complexity. And so then we can do experiments where if we take that simple lipid system and try to recreate the charge density that's in that lipid system that we have in the 8-lipid system, so we increase the amount of phosphatidylserine, we can see a little bit more of that complexity, but it's still not showing the slower component motion that we see. The D2, which is slower diffusion, is much higher in the 8-lipid system than even in a simple lipid system with a lot of charge in it. So even with a lot of charge, you could imagine those negatively charged lipids might be grabbing onto those positively charged lipids in the protein. That might be constraining its motion. That's not sufficient to get the real complexity of the motion. And then also, it's not just one lipid that's contributing to this mobility, this complex mobility, because even though when we take the cholesterol out of the system, and so we now have a 7-lipid system, and you can see with the atomic force microscopy that you don't have the phase transition, you don't have that organization of the lipid system like you do with 8 lipids, you still see 3 components. So there's an ensemble effect of all of these different lipids together that is contributing to this complex heterogeneous motion of RAS. So at this point, we know that there is some contribution from electrostatics. We know that the phase separation, at least in this 8-lipid system because of cholesterol, is not responsible for the complexity of the mobility. But there might be other things that we need to add into the system to really get a full picture. So for instance, we could put actin and polymerize actin onto our system and sort of recreate some of the structure that is found in human cells and see if that affects the mobility of RAS molecules. So I've shown you a variety of approaches that we're using. We're using both cell biology, we're using high-resolution imaging techniques like atomic force microscopy. We also use other techniques like fluorescence lifetime correlation spectroscopy. And then we're also using simulations in order to understand our system better. And through all of those things together, we're getting a more complex and more complete picture of RAS behavior in the membrane, which is really ultimately very important to understand if we're going to be effective in targeting RAS. So sort of linking the first part of my talk to that, the idea we're looking for inhibitors, we're looking for tool compounds, we're looking ultimately for a drug that is going to target RAS very specifically, and understanding how RAS behaves in the plasma membrane and its interactions could give us some important insights and new hypotheses to test and new targets to explore with small molecule drug discovery. Okay, I mentioned at the very beginning that we do a lot of team science. I just want to emphasize that again. Maybe some of you might come to Frederick someday. I know that Rodrigo is coming to do a postdoc when he graduates. But I really hope that all of you would consider Frederick as a possible place to come and do science. We are always looking for people who are interested in working hard and generating new ideas and working on a problem collaboratively as part of a large team. The imaging group up there is listed, Debanjan, Dee, Rebecca, John, Megan, Karen. They're fantastic people, but they're just a small part of an even larger group of people, 50 to 60 scientists working together. And there's a lot of interaction that goes on. So thank you very much for your attention and your time.